Hello, dear students. I welcome you all on behalf of Sri Jendra Rangjun classes. And dear student, we are in a continuation of the chapter that is reproduction in organism of class 12. Dear student, in previous class we have already studied about the lifespan. What is lifespan? How it is correlated with the different characteristics. What are the different types of lifespan of different organisms, that all we have discussed. But after lifespan in this chapter, today we are going to deal with the reproduction and its type. First we are going to understand what is reproduction, then we will discuss the types of different reproduction. So if we go on talking about this, then first we discuss what is reproduction. So, dear students, the offspring grow, mature, and intend or produce new offspring. You know that. If we talk about any organism is going to take a birth, then he is going to grow, he or she is going to grow, and then later on they will be also able to produce the new offspring. So the strategy for this which is used, that is the reproduction, and the reproduction is the only thing that enables the continuity of species, generation after generation. Means we have evolved several years before, but today we are as existing as a human being. So there are so many ancestors of us, and due to the reproduction strategy, the new offspring have developed, and it keeps going on. And due to this, we are here, and every different organism are here. So the first question is, what is reproduction? So reproduction is defined as a biological process of self-perpetuation. It is a biological process of self-perpetuation. Means we are going to continue our genes, we are going to continue our species for the several years, and in which the organism give rise to the new ones or the young ones that are known as offspring. So this is the reproduction. Now, if we talk about this reproduction much more in detail, then we come to know that different organisms have evolved their own mechanism or different mechanism for the evolution. For the what particularly evolution. And every organism has separate mechanism for the multiplication and to produce offspring. But if we talk here, then it is known that the reproduction is classified into two types mainly. One is asexual reproduction, and another one is the sexual reproduction. Now, when we talk about this asexual and sexual reproduction, the bases which are used means it is based on two things. Number one, that is habitat, and number two, that is internal physiology. The habitat and internal physiology. And these are the two things of organism, and several other factors are involved, but these are the two major factors which collectively responsible for how organism reproduce. And that are two means, that is asexual and sexual. So There are two types of reproduction, asexual reproduction and sexual reproduction. Now, when we first talk about the asexual reproduction, what we understand? So here, the offspring is produced by a single parent and with or without the involvement of gamete formation. See, here single parent is involved. Means this asexual reproduction is uniparental. And here another thing is with or without the involvement of gamete formation. It is with or without the involvement of gamete formation. So either gamete form or either not. If you see about the parthenogenesis, then in that the particularly gamete are formed, okay, but fertilization will not occur. So because of this, that is also one particular type of asexual reproduction. So in this mainly what, how we differentiate asexual from the sexual reproduction? In asexual reproduction, dear students, gamete can form, but the fusion of gamete cannot occur. The gametic fusion is absent. And you can see these are the bud which are developing into the plant due to the direct vegetative propagation. Now, if we talk about the parent bacterial cell, it is dividing and it is converting into the two, then it is also a particular asexual reproduction. So, dear student, we will discuss this different type of asexual reproduction, but mainly asexual reproduction differentiated from the sexual one based on the gametic fusion. And in majority books, it is written that it is without gamete formation. But be remember, NCRT says that there can be a possibility of gamete formation. Okay. Now, when we talk about a sexual reproduction, offspring is produced by two parents with the involvement of gamete formation. In this, two parents are involved. And in these two parents are involved of opposite sex means they are biparental. One male and female is involved, and in this gamete formation is also involved. So the particularly sexual reproduction is biparental usually, but can be uniparental. We remember why it can be uniparental. Suppose we take an example of the self-pollinating flower or bisexual flower. So in bisexual flower, the parent is one, but sexual reproduction still occurs. So that is the example of uniparental. Okay. So in this we can write an example of bisexual flower. Now, after this gametic fusion is present, and gametic fusion occurs, male and female gamete fuse and form a zygote. So you can see this diagram is showing these two gametes are getting fused, and after fusion of these two gametes, they are going to form a zygote. So this is a sexual reproduction. Now, first, these are the few things which we have discussed and how the asexual and sexual reproduction can be differentiated. I want to make you to write the more differences between asexual and sexual reproduction so you get it clear. So what we have studied? Asexual reproduction is uniparental. Here, sexual reproduction is Biparental usually can be uniparental, and in uniparental we have discussed about the example of bisexual flower. Also we can take an example of hermaphrodite organism. Apart from these, the important thing is here with or without gamete formation, but here in sexual reproduction gamete formation occur. Apart from this, if we talk about asexual reproduction, then in this gamete are not going to fused. So gametic fusion is absent. Here gametic fusion occur. So zygote is formed. Here in asexual reproduction, it is known that it is quick, rapid, and simple, but it is slow, complex, and elaborate. Apart from this, if you see, then in asexual reproduction, it is by formation of specialized or unspecialized structure, whereas it is by formation of specialized structure, because gametes are a specialized body. But in asexual reproduction, if we talk about the binary fission, in this there is no formation of specialized structure. But if we talk about the spore formation, then it is a specialized body. So here it is by formation by a specialized structure like spore, and here it is by specialized structure that is a gamete. Okay, so in this way, the asexual and sexual reproduction can be separated. Now we are going to talk about one by one asexual and sexual reproduction in detail. So let's, before understanding about the different asexual and sexual in detail, first we talk about some of the MCQs, and easy MCQs we are going to discuss. Lifespan is not correlated with, yes, we have discussed in this previous class, it is not correlated with the size and complexity of an organism, because we have taken example of the parrot and a crow. Both have a same size, but crow have 15 years of lifespan and parrot have 140 years of lifespan. Means if size is small, we can't say that their particular lifespan is of short years or only few years. But if and also we can't say that if size of an organism is large, then the lifespan can be more. Okay, no relation like this of lifespan with the size of an organism. Which of the following has the shortest lifespan comparatively? So it's the banyan tree, royal fern, and banana tree, and moss plant. Actually, moss plant is given because banana tree, banyan tree is approximately having a lifespan, if we say. So banana tree having a 10, 25 years approx, banyan tree having a 200 to 300 years. Okay, and if we talk about royal fern, then it is also having 100 plus years, but most plants do not have that much lifespan. Then lifespan of rice is, if we talk about rice lifespan, then it is 3 to 7 months is given here, or we can say 3 to 4 months. Somewhere data in some books 3 to 4 months is given, and CRT 3 to 4 months it follows. Here it is 3 to 7 months is given. Now the fusion of gametes occur in. So if you talk about the fusion of gametes, it is occur by the in a sexual reproduction. So here the fusion of gamete is going to occur in sexual reproduction. So now we are going to deal with asexual reproduction in detail. Dear students, be remember we are going to talk about this asexual reproduction in detail. In this, in later part, we will also talk about vegetative reproduction. That is also part of asexual reproduction. So first, asexual reproduction, if we talk, we have studied this all things. Single parent is involved, asexual reproduction. And here, yes, one point we have missed during this. The identical copies are produced in this asexual reproduction. The offsprings are identical to each other, exact copies of their parent. Identical. Okay. Now, the question is, in the identical parents which are offspring are produced, this offspring are exact copies of parent, then this also means what? Clone. So plant produced vegetatively or asexually are called clones because they are going to be identical copies. Now, asexual reproduction is common among single-celled organism like plant, a single-celled organism, plant and animal. We have discussed this. Now in this, we are going to discuss one more thing, that is somatic reproduction. What we are going to discuss? Somatic reproduction. Now, in the somatic reproduction, the propagules are formed from the somatic body or from the somatic cells of the parent. And somatic cells are the what? Non-reproductive cells. So if the reproductive propagule is non-reproductive in origin, then we can call it as a somatic propagule. And if the reproduction occurs from that, then it will be considered as somatic or somatogenic reproduction. So asexual reproduction by formation of fission or budding of somatic cells. And this asexual reproduction is going to occur by fission, budding, sporulation, fragmentation, regeneration, and vegetative reproduction. And we are going to discuss that in detail. Okay, I hope you all are getting this. Then, first type of asexual reproduction which we are going to discuss, that is binary fission. Dear student, as the name suggests, binary means in two. So, in this process, parent organism divide into two halves and each half forming an independent daughter organism. So, what here happens? The parent organism divided into two halves and each half form an independent daughter organism. It means the parent body as a whole form a reproductive unit. Means a single celled parent body itself going to act as a reproductive unit. And the parent continue to live its life as a two daughter individual. So the binary fission is exhibited by many single-celled organisms, and the single-celled organism, in case of prokaryotes, they belong to monerans, and in case of eukaryote, mainly they belong to protists. So the example which we are going to discuss, that are bacteria, euglena, and diatoms. Bacteria is a prokaryote, euglena, diatoms are a eukaryote, but all are single celled, so they can exhibit this binary fission. And in this, the particular binary fission, what is going to happen? The parent cell is here. First, the DNA replication or nucleoid replication will occur. You can see the nucleoid or DNA is this much, and now it is replicated. After replicated, the division of cell wall and plasma membrane division occur. And after division of this, they get into the daughter organism. And in this daughter organism, these two are identical to each other as well as they are identical to the parents. So here they start living their life as a two organism, and it is known that organism which undergo binary fusion, that is either prokaryote or eukaryote, monorems or protists, they are considered or they are said to be immortal. So binary fusion in bacteria is given here. Now here, this is binary fusion in amoeba, and here in this, you can see this that this amoeba, the nucleus elongation is occurring in this, and this nucleus get distributed into the two part, and then the cytoplasm get also distributed. Now this is a binary fusion, but it is a simple binary fusion in case of amoeba. And this simple binary fusion is also known as asymmetric or irregular or asymmetric binary fusion. But whereas if we talk about another type, that this is Euglena, and Euglena longitudinal binary fusion occurs. In Euglena, longitudinal binary fusion occurs. And Euglena, if longitudinal binary fusion occurs, the plane of division passes to the longitudinal axis. See, the plane of division passing like this. So it is get distributed into this two. So this longitudinal binary fusion is seen in case of the Euglena. Whereas when we talk about another type, that is in case of Paramecium, that is transverse binary fusion. And in this transverse binary fusion, you can see that the plane of division is transverse, means plane of division run along the transverse axis of the individual, and this transverse binary fusion is found in case of the Paramecium, in case of the diatoms, and in case of the bacteria as well. So this is the transverse binary fusion. Okay. Now, apart from this different type of binary fusion that we have discussed, next the process which we are going to talk, that is budding. Now in budding, which is usually observed in case of yeast, the division of cytoplasm is unequal because the bud which is going to develop, that is small in size, and that small bud develop as a protuberance extension, and that remain attached initially to the pencil. So it is going to be developed as a what? Protuberance or a small bud. So that you need to remember. Now, later on, the bud, the protuberance, the extension is going to separate, and that will going to separate into the new yeast cell. Now, you can see this as a diagram, budding in case of yeast, and here you can see the small bud is developing. So it is a protuberance, it is an extension. Here also you can see this developed bud is going to develop and form a increase in size, and it will in future after maturation look like as a new yeast cell. So if we talk about this budding, it is budding, the division is unequal, is occurring. Now this budding is known to see in case of the different examples. One is yeast, and apart from this, it is also found to be seen in case of Hydra. Okay, in Hydra also bud develops. Then, if we talk about the difference of or compare a budding and binary fusion, then we come to know that how this binary fission can be differentiated from the body, and binary fission can be differentiated from the body. In binary fission, equal cytoplasm division, and here unequal cytoplasm division. Apart from this, here in case of this binary fission, here no protuberance develop, but here protuberance develops, and here equal sized offspring are produced, and here unequal sized offspring are produced. Okay, so now let's talk further. Now next, the type in asexual reproduction which we are going to discuss, that is spore formation. So in spore, if we talk, then member of the kingdom fungi and simple plant such as algae reproduce through a special asexual reproductive structure called spores. The most commonly produced structure or commonly produced spore are the conidia and juice spore that we are going to discuss. So these spores are microscopic, and some of them can survive in unfavorable condition as well. So they are playing a role in asexual reproduction. And as I said, the most common produced spores are conidia, sporangiospore, and juice spores. And out of this also, the considered as conidia is most common type of spore in case of fungi, and juice spore are most common type of spore in case of algae. Okay. Now, the juice spore, first we are going to discuss. So the juice spore, it is a haploid spore. See, one more thing that I want to make you understand here. The spore during asexual reproduction, if we talk, these are produced through mitosis. The meiosis cannot occur in case of spore production. So if they are produced through mitosis, these spore are going to be mitospore. And this mitospore are the asexual spore. Okay, now usually it is produced in case of algae and fungi, and they are haploid. So mitospore or asexual spore are also going to be haploid. So now first we are going to talk about the juice spore. And in juice spore, if we talk, these are motile spore and flagellated spore produced through mitosis. See, they are mitospore produced through mitosis, so they are going to be haploid. But they are motile. Why? They have a flagella. Because of this, they are motile, they can move. The parent cell wall break and the juice spores are liberated in water. But the juice spore, if we talk, we remember it do not have a true wall. They are wall-less. They are going to be naked. And the example are Chlamydomonas, and in this, the protoplast specifically, if we talk about Chlamydomonas, the protoplast cell divide to form 8 to 16 juice spores. Now, this juice spore, if we talk a shape wise, they are look somewhat pyramid shape, and in this pyramid shape, the flagella are attached anteriorly, and they resemble the parent cell. And they resemble the parent cell. So if we talk about certain other features about the juice spore, we come to know that they are produced, they are motile spore, but they are produced within a juice sporangia. Juice sporangia is plural, juice sporangia is singular. So they are produced within a sporangia. So we can say juice spore are endogenously produced spore. Apart from this, the cell wall is absent, so they are considered as naked. Cell wall is absent. And the thing is that other important point that they are formed in favorable condition. If we talk about this here, this juice spore can be uniflagellated or biflagellated. And it is known that this biflagellated juice spore are of two types. Biflagellated juice spore are of two types. One is primary juice spore and another one is secondary juice spore. If you talk about primary juice spore, in this, the flagella are anteriorly attached. But in secondary juice spore, the flagella are laterally attached. And if we talk about their shape, the primary juice spore are pyramid shaped, whereas secondary juice spore are kidney or bean shaped. They are kidney or bean shaped. So that is the what a juice spore. Now, after juice spore, just we are going to see the diagram that see this is the parent cell that is of Chlamydomonas, and this whole cell is going to act as a juice sporangium, and within this juice spore are produced. And juice spore look like this. Now another spore which we are going to discuss, that is a sporangiospore. And in sporangiospore, if we talk, then that are also a particular type of spore. But if we differ it from the juice spore, these are the spore which are produced on by the common bread mold. And this common bread mold are non-motile. So the spore which are produced sporangiospore, that are also going to be non-motile because they do not have a flagella. These spores are produced in sporangium, so they are also endogenously produced. They are also endogenously produced. Okay. Produced within sporangium. And these sporangiospore are the asexual endospore. They are the asexual endospore. Okay. So they are also haploid. And these are known to be seen in the common bread bone, that is the rhizopus. And here in diagram you can see on this braid, the whatever the black dot you are seeing, that is nothing but spores of this rhizopus. If you see it microscopically, it look like this. And this is a sporangiophore, means it is a stalk. And over stalk, this is a sporangium, and within this sporangium here you will see the formation of different this non-motile spores. And this non-motile sporangiospore are also are actually thin walled, and they are formed in favorable condition. Okay, so that is the sporangiospore. After this, the next type of spore which we are going to discuss, that is the next type of spore that we are going to discuss, that is conidia. And in conidia, if we talk, these are also the non-motile spore and produced in case of the Ascomycetes fungus, that is the Penicillium and Aspergillus. And specifically in case of Penicillium, these are spores which are produced at the tip of special hyphae or branches called a conidiophore. In which branches? Conidiophore. Here conidiophore, four word is used for the stalk. So the stalk where this particular spore are produced, that stalk is going to be named as conidiophore. Then conidia are asexual endospore. These are also endogenously produced. And later on, when we see the particular diagram of production of spore in Penicillium, it look like this. And this is a sporangium within which the spore is going to be produced. Now here, this is the diagram of conidiophore. Here you can see this is the hyphae, this is the stalk or a style, and this is named as conidiophore actually. And this conidiophore is going to be branched, and this branches which are formed here, that are primary branch, and this primary branch is named as a rami or rami. After this, these are the secondary branch which are known as medullae. And over the secondary branch, you will find a flask shaped structure, that is phyllide. That is sterigmata. So the sterigmata or phyllides are there, for which the spore conidia, conidiospore are produced. And this conidiospore, if you see, you see, they are produced from here. And if they are arranged in like the sequence, you will see that this conidium is young conidium, whereas the basal one are going to be old conidium. And if basal one is the old and younger one conidium is apex, then this arrangement is known as, sorry, sorry, by mistake I have written like this. See, I want to make you correct this. This is the old one and this is the young one because they are produced from here. So this is the young one. And this is the old one. So when the young conidium is present at the base, this arrangement is known as basipetal arrangement of conidium. Now if we talk about the characteristic features of this conidium, we come to know that this conidium are also a thin walled spore. But this thin walled spore is non-motile or non-flagellated. And apart from this non-motile or non-flagellated and thin-walled spore, these are produced exogenously over steric So these are produced exogenously produced over steric. Then sometimes it is seen that they are found to be pigmented as well. And sometimes it is seen that they can be uni or multinucleated. And if they are thin walled, they are formed in favorable conditions. When the conditions are satisfied, during that type of condition, this all the watery content or the food, all these are available in proper way, then that is a favorable condition and in that the conidia are produced. Apart from this, one more type of spore is there that is produced that is known as chlamydospore. This chlamydospore, if we talk, this chlamydospore it is a thick walled, it is defined as thick walled rusting spore. And if it is thick walled rusting spore, means it is formed in unfavorable condition. And in unfavorable condition, it is formed to pass the unfavorable condition. And this unfavorable condition are passed through this thick walled spore, means we can say that these are perinating spore. Okay, so now suppose this is the filament of any fungus, and when unfavorable condition get encountered, this spore is going to be thick walled and other this are going to die. They are not going to tolerate. Sometimes it can be produced here as well. So this is intercalary spore or medial spore, whereas this is terminal spore. This is intercalary spore or terminal spore. Okay. Now, after this chlamydospore, if we talk another type of spore, yes, this is the chlamydospore. This is the last one. Okay. So whatever we have discussed till now, they are all are the asexual spore. Now, the inner chlamydospore also you should remember that this the intercalary or terminal and chlamydospore is going to germinate and form a new species of fungus. It is known that in bryophytes and pteridophytes also spore produced are haploid in nature, and on a germination they produce a gametophyte that is necessary to complete the life cycle, but that spore in case of bryophyte and pteridophyte are produced from the spore mother cell by the meiosis. They are going to be produced by the meiosis. So if they are produced by the meiosis, they are going to be meiospore. But apart from that, they have some asexual structures like gamete. That gamete is a asexual structure which is found in case of liverworts. In the specific liverworts in which this gamete are found, that is Markensia. And in Markensia, gamete, if we talk specifically, this gamete are produced within gamma cup. They are produced in gamma cup. And this gamete are defined as green asexual bud. And here it is known that one gamete give rise to two new cells. Okay, so this is the part in case of liverworts. So let's talk some of the MCQs related to this, the types, that whatever the asexual reproduction we have discussed. Clones are actually, clones are visible similar in morphology as well as genetically also they are similar, because they are produced through the mitosis, they are produced through the binary fission either, or they are produced through the asexual reproduction. And in asexual reproduction, the identical offsprings are produced, and that identical offspring are known as clones, which are morphologically and genetically similar. Apart from this, asexual reproduction by budding occurs in. So we have discussed that occurs in case of the yeast. Then in which process parent organism divide into two half? Usually in case of binary fission, it divide into two half. Zoospore are produced by, actually zoospore are produced by the algae as well as fungi, and in algae chlamydomonas is more common. And after asexual reproduction and different types, we are going to talk about another type of asexual reproduction, that is a vegetative reproduction. And in this vegetative reproduction, we are going to discuss that this vegetative reproduction also does not involve meiosis and the fusion of gametes. Therefore, due to the absence of meiosis and fusion of gamete, this is a type of asexual reproduction. So let's talk about this in detail. So therefore it is considered as an asexual reproduction, as I told you. Now, in vegetative reproduction, new plants or individuals are produced from the vegetative parts of the plants. Vegetative means non-reproductive. So if we are taking any non-reproductive part of any plant and it is able to form a new organism, now the newly formed individual are genetically identical to the parent plant. Newly formed individual are genetically identical to the parent plant. So they are known as clone here also. The common method of reproduction in flowering plant is this vegetative reproduction. And we are going to discuss this vegetative reproduction in detail. Now this vegetative reproduction occur by two methods, or two types of vegetative reproduction is known. One is a natural method of vegetative reproduction and another one is the artificial method of vegetative reproduction. So first we write this. after natural, there is also artificial method of vegetative reproduction. First, we are going to talk about natural methods. And in natural methods, somatic part of the plant detached from the body of the mother or a parent plant. And after detaching, after the separation, it develops into a new independent plant under the special or suitable environmental condition. Then this process is called a fragmentation. This is detachment and then development into the new one. Then it is a particularly a fragmentation, but it is also helping this. Some plants have specialized structures for the reproduction via fragmentation, and that include the gamma-invertebrates as we have discussed. The detachable somatic part that function in vegetative propagation is called a vegetative propagule. Now, this vegetative propagule, which is a detachable, and this detachable part should have some important thing that help in vegetative propagation. So they can have a one or more bud. Now first we discuss what do you mean by fragmentation. Fragmentation is a form of asexual reproduction or cloning in which organism is split into fragment, and that is by the natural means, any particularly mechanical thing. By any injury they can be cut down into several fragments, and each fragment has capability to develop into the new organism. The vegetative propagule, if we consider, they can be so many. But before that, I want to make you understand that this vegetative propagule, either usually they have a bud, natural bud occur over the node of the stem, and if the particular bud containing part, when kept in a damp soil, this bud is going to sprout and produce a new plant. So some propagules carry adventitious root as well. So we will discuss in different types, and the given following vegetative propagules are tubers in case of potato, rhizome in case of banana and ginger, bulb of onion and garlic, runners in oxalis and grasses, offset we will study in case of Icornia and Pistia. Then we will discuss stolon in nerium, rose and jasmine. We will discuss bulbil as well. Then we are going to discuss the reproductive leaves in case of bryophylla. Okay, so these are all are the vegetative propagules, and they help in vegetative propagation in case of this part. Now, vegetative propagule can be stem, root, or leaf because they all are non-reproductive. So first we talk about the stem, then we will talk about the particularly leaf and root. So now first we talk about potato in tuber and rhizome in ginger. Now dear students, if we talk about the tuber in case of potato, first we need to understand what is tuber. So what happens? This is an underground stem modification. Now see, these all are underground stem. In potato, if someone asks, we will answer that the what is edible in case of the tuber? Potato underground stem. So this is the underground stem. This is the branch. And if the tip of branch going to swell, then that is going to be named as tuber. And in this you will find several notches, and on notches you will find the presence of axillary bud. You can see here. This is the notch. And here you find the presence of axillary bud. This axillary bud will develop and form a new branch like this. Okay, so that is a tuber in case of potato. Another is the rhizome of ginger. See, if we talk about apart from potato or apart from tuber, another underground stem modification which help in vegetative propagation is rhizome. And rhizome is known to found in case of ginger. If we talk about rhizome, this rhizome grow horizontally. First thing. Apart from this rhizome have a internode, node, bud, scaly leaf, and adventitious root. And here the example of this underground stem modification which help in vegetative propagation, that is rhizome, are one is ginger and the other several types are there. Now in this tuber you also write one important another example, that is artichoke. Okay, we will discuss this rhizome in detail now. Now if we talk about rhizome, this rhizome is of two types. Rootstock and straggling. Now rootstock is unbranched and straggling is branched rhizome. Now in an unbranched the example is Bryopteris, which is also known as asperidium. In this, remember one thing, what happens? The underground stem first grow horizontally, then grow obliquely, and in obliquely it grow like this and try to reach the surface of soil. Then this is unbranched underground stem is known as rootstock. Now in this it is of two types. Uniparous sorry here it is of two types, racemose and uniparous zymose. The racemose is known to be found in case of lotus and saccharum, and uniparous zymose, if we see, it is found in case of banana, turmeric, banana, turmeric, ginger. Okay, in all this you will find this particular rhizome. So this is underground stem which help in vegetative propagation. Apart from this, we are going to discuss this bulb and core. So third underground stem modification which help in vegetative propagation is corm. Now corm is somewhat similar to the rhizome, but the difference is it grow vertically. And this corm grow vertically beneath the soil and it bear node, internode, bud and scalings. And if we talk about the example of this corm, the examples are Amorphophallus, and the Amorphophallus is also known as जमीन का. Apart from this, crocus, colocasia, gladiolus, these all are kept. Okay, these all are the examples of corm. And we will see the diagram. See, this is the corm of colocasia here. We will discuss bulb as well, but see, this is the corm. This is growing vertically. It has a node, internode, scale leaf. These are the daughter corm, and they have adventitious root as well. And it is also underground stem modification. And if it has a bud, it can help in vegetative propagation. Now, after this, we discuss bulb. This bulb is also underground stem modification, but in this, the bulb is the stem, but this stem is highly reduced, and this highly reduced disc-shaped stem is known as bulb. This bulb is unbranched, and here the bud is surrounded by, bulb is surrounded by scaly leaves. Now, in this, if you see the onion, I hope you have seen the onion. In onion, internal some leaves like structure are there which are edible, which we eat, and outside the outermost peppery covering is found. Usually we remove that, then we use in our as a vegetables. Okay, so that peppery covering, dry peppery covering, is known as tunic. So this tunic can be there or not. So based on this, bulb are divided into two parts. One is non-tunicated or scaly bulb, and another one is the tunicated bulb. This non-tunicated or scaly bulb, if you see the example, that is lily, and in tunicated bulb, the example is simple tunicated bulb, and in simple tunicated bulb, the example is onion. Because in onion only single outer covering is seen of tunic, but the another type is compound tunicated bulb, and in compound tunicated bulb, example is garlic. So if we want to visualize them, let's see the diagram. And here in this diagram you can see here, this is the bulb. I want also you to make some beautiful diagram here. I want to make you understand everything about this. The onion, the scientific name is Allium cepa. In onion, I hope you have seen that one disc-like structure is found here. Then onion is look like this. Okay. Actually, this disc-like structure is the bulb, a reduced, highly disc-shaped stem. Other part which are found here that are leaves, and these leaves are the fleshy leaves. They are edible leaves, but the outermost is tunic. And in garlic, if we see, you know that there is a presence of one stalk-like structure, and some bulb are present like this. And in this, these are bulblets. Okay, and this is disc-shaped bulb. Okay. Now, in these bulblets, if you see, in these bulblets, if you see, the peppery covering is found over this covering. And this peppery covering is tunic. But this bulblet is actually leaf, and that is edible in case of garlic. Okay, so these are the underground stem modification like tuber, like rhizome, like corm, like bulb, and this all help in vegetative propagation because they have a bulb. Now, apart from this, we are going to discuss some of the creeping stem which help in vegetative propagation. And if we talk about this creeping stem, which are found to be growing attached with the soil. And in that, we are going to discuss the first one, that is runner. Now, in runner, what is seen here, the elongated prostrate subaerial means it is attached to the soil branch. Prostrate means it is going horizontally, it is somewhat elongated, and it is not completely aerial, it is near to the soil or on the surface of the soil, and it is found associated with the or attached with the soil, and it is a subaerial branch with long internode. And you can see this is the grasses in which this runner is seen. So if this elongated branch is growing through the apex, and if it has a node here, this node will give rise to the leaves outside. And here, roots up here. Here, roots up here. So this runner is seen in case of grass. After this, the next creeping stem or subaerial stem which help in vegetative propagation that we are going to discuss, that is stolon. And if we talk about a stolon, here this is the plant, and the plant forming a long lateral branch. So this is the lateral branch, And this lateral branch arising from the base of the stem, and it is first growing obliquely upward, and then it is bending downward and attaching to the ground surface. And when it attaches to the ground surface, the region which has a node that give rise to the new plant and they develop a root. So this obliquely upward and obliquely downward creeping stem or a lateral branch is known as stolon. And the example of this stolon, if we see, they are strawberry and Vallisneria. Okay, so this way a stolon is found. Now, after stolon, another creeping stem which is known to sing, that is offset. And when we talk about this offset, it is a short lateral branch. See, it is a short lateral branch. So it is a short horizontal branch, and this short horizontal branch is one internode long, and it produces cluster of leaves, group of leaves and tuft of root, group of leaves and tuft of root at node. You can see here is node is there where a tuft of group of leaves is above and the group of root are below, and this is a short horizontal branch which is connecting these two, that is known as offset. And this offset is known to found in case of pistia, and another one is the eichhornia. This eichhornia is known as water hyacinth. This eichhornia is known as water hyacinth. Okay, and this water hyacinth eichhornia is known as terror of Bengal. So, if we talk about this, the plants produced vegetatively are clone. And if we talk about eichhornia, it is an aquatic plant, commonly called terror of Bengal. And this is also known as water hyacinth, and which is the most invasive weed which grow in standing water. Dear student, you should remember this. It is the most invasive weed growing wherever there is a standing water, means it is preferred to grow in standing water. And this eichhornia, when grow in a water body, it drains the oxygen from the water, and due to the unavailability of oxygen or a deficiency of oxygen in the water body, which that leads to the death of an organism. Now this water hyacinth, if we see, was introduced in Bengal, and because of its beautiful flower and shape of leaves, but due to its invasive nature, it is turned out to be highly invasive aquatic weed that not only spread to the water bodies of Bengal but also throughout the India. As we have discussed, it drained the oxygen, death of fishes and other animal occurred. And it is very difficult to get rid of them since it can propagate vegetatively by the offset at a phenomenal rate and spread all over the water body in a short period of time. So this is why it is too important. Okay. Now after offset, we are going to talk about next sucker and leaf of bryophyllum we will discuss also. Now what happens in case of sucker that we discuss? In sucker, the main stem grow underground and the main stem have auxiliary bud from this one lateral branch originate that slowly comes above the soil and that developed a leaf like structure, and with that it developed an adventitious root. Now this stem initially absorbs the nutrition from the parent plant. Because of this it is known as sucker. Okay. So the auxiliary bud of the basal portion of the stem or the underground part of the stem form the sucker, which is a lateral branch, creep below the surface, grow obliquely upward, produce a new shoot. And this is seen, sucker is seen in case of the chrysanthemum as well as it is also seen in case of pineapple and banana. Now apart from this, the leaf bud are found in case of the bryophyllum. So till now we have discussed the what particular stem modification, and we have also discussed some of the particularly sub-areolefic stem. Okay. Now in these two things is remaining, then we will discuss the leaf actually of bryophyllum. One is bulbil, then I will come to the bryophyllum. Now this bulbil, if I talk, this bulbil are nothing but they are the large sized fleshy bud of terrestrial plant. And this large sized fleshy buds of terrestrial plant can produce new plant. And this bulbil as given in agave, they are found in agave. There are some another example are there like oxalis, ananas means pineapple, Dioscorea, lily. Okay, in all this bulbil are found. And apart from this, the another one which is found, that is turions, that is also stem modification which help in vegetative propagation. And these are the large sized fleshy bud in aquatic plants. And this also help in vegetative propagation. Okay, so if we take about this particularly examples of these turions, these are found in Utricularia and Utamojaton. Utricularia and Utamojaton. Okay, in that these are found. So we have seen the particularly bulbil. This is the turions. Now after turions, we are going to talk about these all are the what stem modification which are involved in vegetative propagation. Now after this I am going to tell you, or we are going to discuss, that how leaves particularly help in vegetative propagation, and in that we are going to discuss this bulbil, this leaf buds. So the given example of leaves which are helping in vegetative propagation. So what happens in case of this? See, this is the leaf of the bryophyllum. In the leaf of bryophyllum, they have some adventitious bud here, and this adventitious bud grow and help in vegetative propagation. So when this leaf with the adventitious bud reach on the soil, they will absorb the root nutrition from some of the developing roots, and that will develop a new plant plate like this, and that will develop at the margin. And if this is developing at the margin as a new plant, that is going to form a new plant, means helping in a vegetative propagation. So the vegetative propagation through leaves are found in adiantum, that is walking fern, then begonia, okay, calonche. In all this, the leaf help in vegetative propagation. So this is all about the particular modification which help in vegetative propagation in case of the leaf. And we have discussed stem, we have discussed leaf vegetative propagation naturally. Now we are going to discuss how root help in vegetative propagation. So we know that both taproot and adventitious root take part in vegetative reproduction actually, and that is due to the presence of bud. So bud can present on taproot and adventitious root. Now this bud is known as radical bud. Now this adventitious bud on root detach and give rise to the new plant. Now we are going to discuss some example in which new plant developed from taproot in Shisham, that is Dalbergia Siso, Populus, Cydium, LBGA. Whereas the new plant developed from adventitious root in Ipomia patatas, yam, dahlia, and asparagus. So this way Particularly, root also play a role in vegetative propagation. So, dear student, here the natural way of vegetative propagation is over, and the different types of asexual reproduction are also over. Now we are remaining with the just artificial methods of vegetative propagation that we will discuss in next class, and then we will continue to the sexual reproduction. Till that, thank you.